Hello, today we discuss about the study of the microscope presented by Dr. Kamthane DC, Head Department of Microbiology, SGB College, Purna. Microscopes are the instruments designed to produce the magnified image of the small objects. This microscope accomplishes three tasks. Number 1, it produces a magnified image of the specimen. Number 2, separate the details in the image, and number 3, render the details visible to the human eye or the camera. There are two types of the microscopes. Number 1, simple microscope, and number 2, compound microscope. Simple microscope is the microscope which is having only one type of the lens. The compound microscope is the microscope having the more lenses as eyepiece and the objective lens. The first microscope it is given by the scientist Antony van Leeuwenhoek, while the compound microscope it is given by the scientist Zacharias Janssen. Compound microscope it is having the three essential systems, and these systems are as number 1, support system, number 2, illumination system, and number 3, magnification system. In the support system, the base, pillar, stage and body tube, these are included, while in illumination system, light source or the mirror, iris diaphragm and condenser it is included. In magnification system, objective and eyepiece are present. This is the diagram of the microscope. This microscope appears externally as in the left side figure, while the labeling it is done in the right side diagram. The first, it is the base, and this base it is of horseshoe shaped. It supports the whole microscope. Above this base, there is the pillar. This pillar it is joined with the base with the inclination joint. Above this pillar, the arm it is present, and at the end of arm on the another side, the body tube it is there. Above this body tube, the draw tube it is present, and this draw tube holds the eyepiece lenses. Below this draw tube, body tube it is present, and this holds the revolving nose piece. The revolving nose piece it is called because it revolves, and on this the 3 to 4 objective lenses are mounted. The lens which is near to the object it is called as the objective lens, while the lens which is near to the eye of the observer it is called as the eyepiece lens or the ocular lens. Then these objectives are present. Below this, the stage it is there, and this stage supports the slide which is to be examined. Above this foot, the mirror it is present, and which adjusts the light which is required for the observer. Above this mirror, the iris diaphragm it is present. This iris diaphragm it is having the leaves. This maintains the intensity of the light which is passing from the mirror to the condenser. The condenser it is present above the mirror, and this condenser function is it condenses the light, and this pass the light upward in the form of the pencil shaped shape, and this stage it is having a hole which passes the light transmitted from the mirror to the condenser and then to the stage. On the stage our slide it is present, and this mirror it is present on the slide. Then the total magnification we have to discuss. The magnification of the microscope is equal to the magnification of the objective lens multiplied by the magnification of the eyepiece lens. Suppose if the eyepiece it is the 10x and we are using the low power objective lens, then the total magnification it is the 100, and if we use the high power objective lens having the magnification 40x, the magnification it is 400x. If we use oil immersion lens having the magnification that is 100x, then it gives the magnification that is 1000x. Then working and handling of the microscope. First, put a drop of cedarwood oil on the smear prepared on clean grease free slide. Then keep the slide on the stage. Fix the slide with the clips of the rack and pinion system. Firstly, adjust the light with low power objective, that is 10x objective. Then replace 10x objective by the 100x. Dip the oil immersion objective, that is 100x objective lens, in the oil present on the smear. Then focus with rack and pin arrangement. Then focus the microscopic field with the coarse adjustment knob. And lastly, finally adjust with the fine adjustment knob to observe the field in having the bacteria. We have to take the care of the microscope, and in this always carry the microscope with two hands, one hand beneath the base and one hand on the other end of the microscope, that is holding to the arm. Keep microscope away from the edge, so do not disturb the position of the microscope. Avoid tilting of the microscope when looking in. Do not touch glass surfaces of the lenses with fingers. Always return the objective lens to low power when finished. Do not remove any part of the microscope. Clean the lenses with minimum amount of xylene with muslin cloth, and keep the microscope free from dust in a separate cabinet. Thank you for listening.